biotechnology means biological systems or processes, and technology means methods, systems, and devices used to make useful products from these biological systems. In biotechnology refers to the different technologies that make use of living cells or biological molecules to generate useful products for the benefit of mankind. In biotechnology involves knowledge sharing between different areas of science, such as cell and molecular biology, to generate useful biology for the benefit of mankind. Modern biotechnology involves knowledge sharing. It combines cell and molecular biology, biochemistry, and computer science, all through recombinant DNA technology. Ancient biotechnology had taken root as early as in the Paleolithic era, around 10,000 years ago, when early farmers began to cultivate crops. The most classical example of biotechnology, as early medieval times, is the use of fermentation technology for production of bread, cheese, wine, and beer. The practice of making fermented food, such as dahi and idli, kinema, and beverages, using local biological resources, was common in medieval India. The relevance of traditional Indian knowledge in making curd, known as dahi, has been indicated in few of the patents found in the United States Patent Database. Fermentation can be explained as a microbial process in which enzymatically controlled conversion of organic compounds occurs. It was practiced for years without any actual knowledge. Fermented dough was discovered by accident, when dough was not baked immediately, and consequently, it underwent fermentation by yeast such as Saccharomyces winlocki. The 19th century witnessed an increase in the production scale of immediately fermentation-based products such as glycerol, acetone, butanol, lactic acid, citric acid, etc. Modern biotechnology is based on rDNA technology. It allows scientists to cut and join pieces of DNA and place the new recombinant DNA into a host. Since the advent of rDNA technology, biotechnology has led to advancements in medicine, agriculture, animal science, and environmental science. Modern biotechnology is based on rDNA technology. It has a wide range of applications, like therapeutics, diagnostics, and crop improvement, and even development of biodegradable plastics. Modern biotechnology focuses on 4 major areas: medicine and healthcare, crop production and agriculture, food processing, and environmental protection. Using RD and tools of bioinformatics, modern instrumentation, and bioprocess, synthetic drug analogs can be predicted and possibly synthesized, which may show improved disease treatment. Using RD and production of vaccines and gene therapy are also important applications of applications of biotechnology in the field of medicine. Using rDNA technology has been successfully applied for the development of biopharmaceuticals with therapeutic value. Different protein molecules, which may act as drug molecules, are being expressed in heterologous systems, such as microorganisms, plants, etc. A number of therapeutic products including antibiotics and hormones, have been produced using rDNA technology, which are available in the market, etc. A common example of therapeutic protein is human insulin, used for the treatment of diabetes, a disease in which blood sugar levels are elevated. Classic example of a human protein, expressed in Escherichia coli. At present, insulin is being produced predominantly in E. coli and Saccharomyces cerevisiae. Classic growth hormone is another example of successful production of desired proteins in different microbial host systems via rDNA technology. The human growth hormone is another example of successful production of desired proteins in different microbial host systems via rDNA technology.
Many human proteins have also been expressed in milk of transgenic sheep and goats. For example, FDA has approved the production of blood anticoagulant in milk.
Currently, scientists are trying to develop such drugs against diseases like hepatitis, cancer, and heart diseases, which are the leading causes of human mortality. Gene therapy technology is most helpful in the treatment of diseases caused by gene defects, such as cystic fibrosis, thalassemia, Parkinson's disease, etc. Conceptualized in 1972, gene therapy involves delivery of required gene into a patient's cell as a drug to treat disease, so that it replaces the function of the defective gene. The first attempt, although unsuccessful, was performed by Martin Klein in 1980 for treating beta thalassemia. Hi everyone! Genetic testing is a type of medical test. It helps in identifying defects, such as chromosomal defects in gene and protein expression anomalies. Hi everyone! The first successful gene therapy was achieved in 1990. Ashanti da Silva was treated for ADA deficiency, an autosomal recessive metabolic. Hi everyone! It helps in determining a person's chance of developing or passing on a specific disorder. Hundreds of genetic tests are currently in use, and many are being developed.
Russia approved neovasculgin in 2011 as a first-in-class gene therapy for peripheral artery disease. For example, genetic tests for phenylketonuria and congenital hypothyroidism have been developed. Phenylketonuria patients lack an enzyme. It is needed to metabolize the amino acid phenylalanine. This is responsible for normal growth.
Transgenic plants harboring these improved traits are good examples of the application of biotechnology in agriculture. Conventional plant breeding techniques have improved varieties. They have not been able to keep pace with the increasing demand for food, vegetables, and fruits. Use of rDNA technology has successfully led of a number of transgenic plants exhibiting resistance to pathogens, salt.
In these transgenic plants, useful genes have been stably incorporated into the plant genome, which resulted in the stable expression of targeted gene product. Transgenic plants have been developed for production of edible vaccines by expressing antigenic proteins to retain its immunogenicity. Individuals are expected to be immunized by simply consuming such transgenic plants. Potato-based vaccines against measles, cholera, Norfolk virus, etc.
Biotechnology played a major role in agriculture by facilitating genetic manipulations of important crop plants to develop biotic and abiotic stress resistance plants. The 5 major traits used for crop improvement are insect resistance, herbicide resistance, virus resistance, delayed fruit ripening, and nutritional enhancement. Biofuels are produced through biological processes, rather than a fuel produced by geological processes such as coal and petroleum. Biofuels can be derived directly from plants, or indirectly from agricultural, commercial, and industrial wastes. Basically, it involves generation of biomass that can be converted to convenient energy-containing substances via different ways such as thermal conversion.
This biomass conversion are in solid, liquid, or gas form. The major types are bioethanol, produced via fermentation of sugar and starches by microorganisms. This biobutanol, a biofuel, is often a direct replacement for gasoline. Biodiesel is the most common biofuel in Europe, produced from oils or fats using transesterification. This first commercial-scale plants to produce biofuels from cellulosing organic matter, operating in the United States. In parts of Asia and Africa where dry lands prevail, sweet sorghum is.
Biotechnological tools can help in improving the edibility, texture, and storage of the food, prevention of mycotoxin production, extending shelf life. Protein engineering of microbial enzymes are produced commercially at a large scale by culturing the microorganisms in tanks and industrial-scale fermenters. Industrial-scale production of fermented foods has also been made possible, such as cheese, yogurt, certain probiotics, other popular fermented products.